Western blotting, also known as immunoblotting, combines the superior resolving power of SDS polyacrylamide gel electrophoresis, or SDS-PAGE, with a highly sensitive antibody binding assay to detect a specific protein in a mixture of proteins. Proteins are first separated by size, by SDS-PAGE. Detecting a specific protein by SDS-PAGE alone is usually not possible. Western blotting provides the sensitivity to detect a specific protein. In this technique, proteins are transferred from an SDS-PAGE gel to a polymer sheet in the blotting step. Protein transfer from the gel to the polymer sheet is done by electrophoresis. In an electric field generated by a power supply, the proteins, which are coated with negatively charged SDS molecules, migrate toward the positive electrode. As the proteins migrate out of the gel, they are captured on the sheet. The proteins stick to the sheet in the same positions they had in the gel. Thus, the sheet is an exact replica, or blot, of the gel. The empty gel is discarded. Then, a primary antibody that will only bind to the protein of interest is incubated with the blot. After a washing step, only the primary antibody that is bound to the protein of interest remains. Several methods can be used to detect the antibody-labeled protein. All of these methods use a secondary antibody, which binds specifically to the Fc domain, or the tail region, of the primary antibody. Typically, the secondary antibody is linked to an enzyme that produces a chemiluminescent or colored product, or it contains a fluorescent tag, enabling identification and quantitation of the protein of interest. Illumination with the appropriate wavelength makes the tag fluoresce. The emitted fluorescence can be detected by specialized detectors. Western blotting makes it possible to find a protein in a complex mixture, the proverbial needle in a haystack.